everybody. My name is Saima. I'm an undergraduate researcher working under Dr. Howard. So before I start my presentation, I'd like to tell you a little bit about myself. I'm actually a biology major, and I'm pre-med, which is not the kind of person that you usually see working in labs, but I always wanted to get involved in research because I always found it to be a rewarding experience. I did a lot of research in high school. So when I came to UT and I realized that it was an amazing research institution, I thought that this was an opportunity that I couldn't let slip away. So I started looking into the different projects that were going on in UT, and Dr. Howard's project really stood out to me because it was so fascinating to see that something that I had considered a medical problem, genetic epilepsy, has so much of a background that you really have to understand and think about before you can start talking about diagnoses. So that's how I got involved, and now I'll start my presentation. So today in my presentation, I want to start with an introduction to Dravet syndrome, which is the syndrome that we work on in the lab. And then I want to tell you a little bit about Howard Lab, and then a little bit about my work, and then the impact that the Needleman internship has had on my work. So Dravet syndrome is a severe form of epilepsy that manifests in infants and young children, and it's categorized by prolonged and frequent seizures, and particularly febrile seizures. It's also characterized by developmental and behavioral delays and things like motor deficiencies, heart problems, and a whole bunch of consequences that come with it. Also, something that's really terrible about this epilepsy is that it has an extremely high mortality rate of around 15 to 20% from SUDEP, which is sudden unexpected death from epilepsy. And treatment options are limited, and so there hasn't really been a viable option to kind of stop the epilepsy. So that's kind of what we're looking into. So let's talk about the science behind Dravet syndrome for a second. So Dravet syndrome is strongly associated with mutations to SCN1A, which is a gene that codes for the protein NEF1.1, which is a subunit of the sodium ion channel. And so there's a new field of research that also links Dravet syndrome to a mutation in SCN1B, which is a gene that codes for an auxiliary subunit of the ion channel. So it's not a direct subunit, it's actually just kind of like a helper subunit, and that one is beta-1. And the sodium channel plays a really important role in the generation and propagation of action potentials, so you can see how a mutation to the sodium channel or to an auxiliary subunit of the sodium channel can have devastating consequences on the human body. All right, so now a little bit about our lab. So Howard Lab is investigating these two gene mutations to gain a better understanding of the effects of their effects on the brain. And this is because a better understanding of the types of cells that are affected can lead to new cell-specific therapies that may be able to actually counter the consequences of having this form of epilepsy. So now let's talk a little bit about my work. I've been in Howard Lab for approximately a year now, and I've been there since the lab had just started, so I helped with setup. I unpacked a lot of boxes in the beginning, and so then I kind of moved on to general upkeep of stock solutions and helping Professor Howard with mouse updates, keeping him informed. And as my role has progressed in the lab, I've started to do perfusions on the mice and extracting their brains and doing brain slices. And now, more recently, I'm working on immunohistochemistry, which is staining the brains with antibodies. And I also do brain imaging. So my primary focus this summer with the internship has been immunohistochemistry. I test all of our incoming antibodies, and I make sure that they work well and that they're easy to use, which, as Professor Howard knows, has been a challenge. I also have started to test our antibodies for co-labeling success. So when we're using the two antibodies together, are there any problems? Do they work well? And currently I'm working with SCN1A mutant brains, which is the primary source of mutation for Dravet syndrome. to see if there's any drastic impact on the specific cell types, such as cells expressing NPY, calretinin, somatostatin, parvalbumin. And this is important because knowing which cells are particularly affected with this mutation can help lead to the development of drug therapies that specifically target only those cells. And that's really great because of two reasons. First of all, it makes our drug therapies more efficient, it makes them better, and also it makes them less harmful for the rest of the brain and for the rest of the body because you're not targeting the whole brain, and so the harmful consequences are reduced. So here's some images that I've taken from my experiment. Hopefully you can see them on there. So all of these are just experiments that I've been doing over the course of the summer, and a lot of the staining is either NPY staining or calretinin staining because I found those antibodies to be working best. And here is my favorite picture from what I've been doing. This is actually a co-labeling with NPY and calretinin antibodies, and this is also like a whole brain image, and what's showing up here is the hippocampal region. So this was a really exciting result that I got. So after the summer, I will be continuing in the lab, and what I hope to do in the future is, first of all, continue staining the SCN1A brains, and then being able to compare the results that I get to wild-type mouse brains so that I can see and quantify whether there is a significant impact on any specific type of cell. And I also hope to learn to do tracer injections, which we've kind of talked about in lab. And lastly, what I think is most important is share the knowledge and the advice and the support that I've gotten through this experience with other young researchers in any way that I can possibly help. So let's talk about the reason that we're all here, the Noodleman internship and the benefits that came with being an intern this summer. So this experience for me was amazing for many reasons. First of all, it provided me with the opportunity to meet a lot of great people who share the same interests as me and have the same ideologies, which was really great. And also the foundation itself has been a great form of support and guidance throughout the summer, all the meetings that we had, and just knowing that your science and what you're doing, even as an undergraduate who may not be doing the most complicated things, just knowing that someone believes in you and someone has your back, that was great, and that gave me a lot of confidence. And also, this experience highlighted to me how important the work that Judy is doing is, because I was thinking about it, and in high school, no one really talked about research as a viable career path. When you're in high school, people talk about you can be a doctor or a lawyer or an engineer or stuff like that, and no one really talks about, hey, you can be a researcher and find cures and do amazing things. So that has definitely been really enlightening, and I think that it's really important to kind of put that into the minds of young people as they're going through their pre-college education. And also, personally, I guess, this internship and working with Dr. Howard has made me realize that I do truly have a passion for research. So my goal in life has always been to become a doctor. But now, like Ryan was saying, I'm starting to see the lines blur. So I think it's important to have both a research background and a medical background if you want to be a successful doctor, because just having those perspectives itself is important. And I'm not sure about what career path specifically I want to take. I've been looking into MD PhD now, which is really exciting. But I know that in some way or form, I want to be involved in research in the future. And lastly, the Noodleman scholarship that was provided for the interns was really great because it made it possible for me to just solely focus on research this summer without worrying about anything else. And that gave me a lot of time to really discover my interests and kind of build up the confidence that I have now. So I'm extremely grateful and thankful for this amazing opportunity. And before I finish, I just want to take a minute to say thank you to Dr. Howard and to Judy, the head of the foundation, for providing me with this wonderful opportunity. It was a really great experience. And I also want to thank everyone that's here today for how you're helping shape young minds through whatever work that you're doing. And I also want to thank Matt for the great work he did with filming. That wasn't easy. The camera he was carrying around was so heavy. I don't know how he did it. And also, I would like to take a moment to kind of express my admiration for Harvey Noodleman. It takes a really special kind of person to inspire the creation of a foundation. And I know that he was probably really amazing, and I wish I could have met him. Thank you.